Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. We have so far discussed all the sequencing technologies that we use today, and in this class we will be talking about some very important technical terms that we will be using throughout this course. Okay. So, these two terms are given here, right. The first one is called sequencing coverage. Okay. So, this is very important for designing any next generation sequencing experiment, and we will be talking about quality score. Okay. So, this quality score, as we will see, is very important for interpreting the data or ensuring the data that we are using for interpretation is good. And finally, we will have some discussion on experiment design. Okay. So, maybe in some point you will have to design an experiment, NGS experiment, right. You want to use these NGS methods for your project or for some work, right. And you might be wondering which platform should I use, what should be the read length, how many reads do I need, what kind of sequencing method should I adopt, right. So, these questions of course require careful examination of different parameters. Okay. So, at the end we will discuss about this experiment design. Okay. So, these are the keywords: coverage, quality and design aspects in this class. Okay. So, just to start with the brief overview of this NGS data analysis method. Okay. So, here is the flow chart. Okay. So, the first step, we have this data from a sequencer, right. Whichever sequencer you use, we have this data. This is quite significant amount of data usually, right. These data sets are huge, and again the data set data format could be different. Okay. So, we will talk about this data format in the next class, because these sequencers give out data in different formats. Okay. So, you will have to deal with these different data formats. Okay. Now, the first step that we have in this data analysis process is the data quality check or QC. Okay. We want to ensure that the data that we have got is of good quality. Otherwise, if we do all the downstream analysis without doing this quality check, the conclusions that we would get might be erroneous or might be wrong. Okay. And this is something we want to prevent, right. So, at the onset, right, we want to do this data quality check or QC. Now, you can take different paths after you have done the quality check and you see you are satisfied that the data is good, then you can take different paths for different types of analysis. Okay. So, briefly, one path that we have is the assembly path, okay, read assembly. So, this is true for new reference genomes, right. So, if you are sequencing a new genome or new organism, right, for which there is no genome data available, the first thing we do is we do an assembly, read assembly, and we get the new reference genome. In other cases where you have a reference genome already, what we do is something called read mapping. Okay. And this read mapping happens against a reference sequence. Okay. Again, these two steps are completely different, right. This read mapping and read assembly, these are quite different, and they have their independent algorithms for doing these steps. Okay. We will discuss this in later classes, this read assembly process, the algorithms, the tools, etc., that are out there. Similarly, for read mapping, we will talk about the algorithms and the tools. Now, once you have done read mapping, again this read mapping is required for a lot of analysis, lot of different types of analysis. For example, if you are interested in genome analysis, right, for example, you are looking for genetic variations or structural variations, right, single nucleotide polymorphisms, you will do this genome analysis, and the first requirement is the read mapping. You can also be interested in measuring expression level of mRNAs, right, or genes, right, and we will do something called transcriptome analysis, and this is the first step is again read mapping. Okay. And then finally, for epigenomic studies also you need to do read mapping. Okay. So, we will talk about these steps in much more detail. Okay. These genome analysis steps, identification SNPs, structural variations. We will have a dedicated section on transcriptome analysis part. We will also talk about the epigenomic studies. Okay. So, the assembly problem we can summarize in this very simple cartoon, right. So, what you get is the reads, right, from the sequencer here on top. Okay. So, the reads are shown in different shades of blue, and what you want to do from these reads, you want to build a single continuous sequence, right? That is the goal. Okay, and this is what we call as the reference genome sequence, right? For example, the human reference genome, right, or mouse reference genome, right? So, we have these DNA fragments, right? We isolate cells, we have these DNA fragments, we sequence using a sequencing platform, we get these reads, and finally, the ultimate goal is to know the full sequence, right? Get the full sequence in one continuous stretch. Okay, this is something what we call the assembly problem. You have something else called the mapping problem, right? As this is associated with the mapping, read mapping. So, what is the mapping problem? So, we got this bunch of reads, and you also have the reference genome sequence. Okay. For example, let us say we sequence a human genome now, right? The reference human genome sequence is available. So, what we will do is we will take these reads and we will start to find out, okay, which part of the reference genome this read comes from, right. We are essentially mapping it back to the reference genome to know the location of the reads, okay, and this will help us identify the changes, genetic mutations that are happening at those specific positions. Okay. So, this is the mapping process, this cartoon shows you this, right. So, for example, the first read we see, okay, this kind of matches with this part of the genome. So, we put it there, right. Okay. We kind of keep track, okay, this read maps here in this genome, and we then go on to the next read, right, and we see, okay, this one matches with this part of the genome, right, we put it there, okay, and we continue this process until we have mapped all the reads, okay, against the reference genome. Okay. So, this is kind of the mapping problem in brief. So, the term sequencing coverage that we use actually comes after we have done the mapping. Okay. So, this is the sequencing coverage, or sometimes we also refer to this as sequencing depth. Okay. So, for each base in the reference sequence, number of reads mapped to that position refers to the sequencing coverage, right. So, this determines the sequencing coverage. Okay. I will just illustrate again with the cartoon, right. So, after this mapping, if we take these 4 positions, right, position A, B, C, D, right. So, for these positions we just check, right, how many reads are there which have mapped to that position. Okay. So, for example, for position A you have just one read mapping to that position. So, you have coverage of 1. For position B you have 4 reads mapping there, for coverage 4, position C coverage 5, position D coverage 2. Okay. So, this is how we calculate coverage for each base in the reference genome sequence. Okay. Now, there is another term which is called the mean coverage. Okay. So, this is mean of coverage for all positions across the whole genome. Okay. So, if you take coverage value for each position of the genome and average, that will give you the mean coverage. Okay. So, here is the mean coverage formula, right. So, this is the coverage I summed over I equals to L, and then of course, you have to divide by the length of the genome. Okay. So, that is something that you have to do, right. So, this is the length of the genome. Okay. So, L is the length of the full genome, right, and coverage I is the sequencing coverage for the base position I. Okay. Now, so how do you represent this mean coverage? So, when you look at genome sequencing projects, sometimes you will see this, right, 10x coverage, right. What does it mean? So, the mean coverage is 10. Okay. So, on average there are 10 reads mapping to each position of the genome. Now, this term on average is very important, right. So, it does not mean that all positions will have 10 reads mapped. Okay. So, this is something you need to be aware of, right. Even if you say, okay, mean coverage is 10x, right, it does not mean every position will have 10 reads. Okay. So, this is something that we observe across this data, variation in coverage. Okay. When you do a genome sequencing project, we do the mapping and we see these fluctuations in coverage across the genome. Okay. And this is observed because you have something called sampling bias and amplification bias. Okay. So, it can happen, right, some positions you have seen these examples, right, 1, 4, 7, and in some cases it might be 0, right, some positions there might not be any read mapped at all. Okay. And then again the coverage could fluctuate between 1, 5, 7, etc. Okay. Again this is something you need to be aware of. Okay. Now, what is sampling bias? I mentioned sampling bias. So, this is actually because of random selection of DNA fragments from the pool, right. So, when you are preparing the library, you are fragmenting the genomic DNA, right, and you are generating these fragments, and only a part of this fragment you take for the amplification or direct sequencing, right. So, there is a bias, right. Sometimes there could be a bias, right, which region, which fragments you are choosing for the next steps. Okay. So, that will be the sampling bias. You can also have something called amplification bias, right. So, many of these next generation methods, right, we have discussed so far, they require this amplification step. Okay. Except nanopore, all of them require this amplification step, and this amplification can select certain DNA molecules, right. So, it can preferentially amplify specific DNA molecules, right, and this happens because there are a lot of factors, but one of them is the GC content, right. So, there is an optimal GC content at which there is a very good amplification. There could be the mismatch, right. So, mismatch will reduce amplification, etc. So, there is a lot of amplification bias that will happen during this library preparation step. Okay. And this is something that will again generate this variation in the coverage. Okay. Now, the question is, why do we care about this mean coverage, right. So, this is very important. So, one of the first point is this has very important implication for designing an NGS experiment, right, and it helps in deciding which platform you want to use and the kind of throughput that you require, right. So, this is the formula that we will use, right, when we are designing an experiment, is that the number of reads that are needed for our experiment is actually given by this. So, if you have if you know the genome size and you know the coverage that you want, right, for example, 10x, 20x or 30x, so mean coverage will be 10, 20 or 30 respectively, and divide by the read length. Okay. So, if you know the read length, because you are going with a specific platform, you know the median read length or mean read length, right, you can calculate how many reads you would need, right, if you know the coverage. Okay. So, again then that will help you choose the right kind of platform, right, whether you need 100 million reads or whether you need 10 million reads, right, depending on that you will choose the platform. Okay. And again you can turn it around and you can actually calculate what is the mean coverage that you will get if you know the number of reads that you will get from a platform and you know the read length of that platform divided by the genome size, right. So, you can play around with this, right, and you can calculate whichever the parameter you need, right, what kind of mean coverage or what kind of read number you would need for your sequencing project. Now, there is a term called deep sequencing related to this coverage term. So, this actually refers to sequencing a genomic region multiple times and where the mean coverage would be 100 or between 100 to 1000 or even more, right. So, it is deep, really deep sequencing, right. So, coverage is 100x, 1000x or even more, right. So, why do you do this deep sequencing? Because we want to identify rare clones or cell types within a cell population. For example, a cell type is present only in 1% of the cell population, right. So, in that case you probably want to go for, let us say, 1000 coverage or even more, right. So, this is kind of an application that we have, right, and the lower limit of detection frequency, right. So, whether you will detect cell type which is present at 1% frequency or 0.1% frequency is determined by the coverage of the deep sequencing experiments, right. Now, coming to the second important term, right, this is a very important term which is called the quality score. Okay. So, this quality score actually is a measure of confidence in a base call by the detector from the signal, right. So, each of these sequencing technologies, right, there is a detector, there is some sort of signal, right, by which the detector detects the bases, right, or calls the bases, and this quality score defines the confidence in that base call, right, and this is actually implemented in the instrument and this is available for all bases that are sequenced in the instrument. Okay. So, this is something we may not be aware of, but we just see the end results, right. How the whole process happens we may not know because it is already implemented in the instrument, but this is there and the data is actually reported, as we will see, right, when we talk about data formats. Okay. Again, this is something kind of measuring signal to noise ratio. There is a background noise and how strong the signal is compared to the background noise, that kind of tells the determines the quality score. And sometimes this quality score is also referred to as Phred score because of the first program that was used is called Phred for the Sanger sequencing based quality determinant. Right. Now, how is this quality score important? So, this quality score gives a probability of error in the base calls. Okay. So, this is something looking at the quality score we can tell, okay, what is the chance that this base is wrongly called. Okay. This is not the right base. Okay. So, you see this formula here. So, the probability of error is given by 10 to the power minus q by 10. Okay. So, where q is the quality score, okay, and then we can calculate this percentage accuracy, which is given by 1 minus this probability of error into 100, right. So, if you are calculating this accuracy in percentage, like we have seen for Illumina, right, 99.9% accuracy, or for other platforms we have seen 90%, 95%, whatever the accuracy is, we can calculate using this formula, right. So, one of the things again you will see is something called this Q10, Q20, Q30, Q40. Okay. So, what does this mean, right? So, you see this, you will see these terms for each platform, right. So, for example, in Illumina you will say, okay, we have like 99% base above Q30, which means they have 99% of the base calls above 30 quality score. Okay. So, the error rate, probability of error is very low, and then you can calculate the accuracy, right. That is how you arrive at this accuracy. Or for example, in nanopore you say, okay, we have achieved Q20. Okay. So, most of the bases now are above this 20 quality score. Okay. So, this is something that is very important, you see this term, okay, because this is used in determining the accuracy. So, just to illustrate, right, if q equals to 10, right, p equals to probability of error is 0.1, which means 1 in 10 error, this is a high error rate. So, you have 90% accuracy. If q equals to 40, so probability of error is 10 to the power minus 4, right, you just use this formula here, right, and this means 1 in 10,000 error, right. So, the chance of a base being an erroneous call is actually very, very low, right. So, higher q value means lower chance of error. Okay. Now, one of the questions that you might have in your mind is that does this quality score apply to homopolymeric stretches, right? Would it accurately reflect the error that happens in homopolymeric stretches, right? For example, in 454 or in ion torrent. Okay. So, in 454 or ion torrent, this length of homopolymeric stretch is determined from one single peak, right. It is not like separate single peaks, separate signal peaks, right. So, that you have these signal peaks coming for each base, right. So, if you look at the quality score of these bases in the homopolymeric stretch, they are actually very close to each other, right. They do not differ so much from each other. So, what researchers have seen that Phred score is actually not informative enough. Okay. So, you cannot just rely on Phred score if you are looking at homopolymeric stretch in 454 data or ion torrent data, right. So, and researchers have developed more complex base calling models, right. We will not go into all those complex models, but what I am saying is that you need to actually be careful in just relying on Phred score in case of homopolymeric stretches, okay, because the detection happens from one signal peak, right, and if that peak is well resolved compared to noise, you might get good quality score. Okay. So, coming to the last part, right. So, we have looked into coverage, we have looked into quality score. The last part is about experiment design, right. Now, when we are starting a project, right, or doing some experiment something, right, and one of the questions that we might have is that which NGS technology should I use for my experiment, right, and this is a question that we always face, right, when we are doing our research, right, we also face this question, right, which NGS technology is suitable for me. Again, there is no single answer, right, it depends on the requirement, depends on your project, right, depends on many factors, right. So, before we go there, I just wanted to give you this overview of this comparison, right, of different NGS technology that we have discussed so far, right. We have 454, Illumina, SMRT, ion torrent and ONT. Okay. And there are a few important points that is really relevant for us. One is the accuracy, right, this is something that we care about. So, 454 gives you 99% accuracy, right, Illumina about 99.9%, SMRT long reach about 90%, let us say, and high fight you can get above 99%. Ion torrent about 99% accuracy, Oxford nanopore about 90%, but if you are using consensus call, you will get above 99%. For read length, right, so 454 we have talked about 500 to 1 kb, Illumina is about 250 base pairs. If you are doing paired end, you get 2 times 250. SMRT you will get above to 3 kb, right. Ion torrent is about 400 to 700 base pairs and ONT is more than 4-5 kb. Running cost, this is a very important point, right, with the funding that you have, right. So, running cost will be 454 was very expensive, so it is not available anymore. Illumina, SMRT, their running cost is on the higher side because of the polymerase and the modified nucleotides that you use. Ion torrent is slightly more economic compared to Illumina, SMRT because you do not use any modified nucleotide. And of course, since we have discussed all the methods, ONT is the most cost effective because it is doing direct sequencing, there is no polymerase or DNTP involved. Coming to real time observation, so 454 Illumina ion torrent, they are not real time observation, whereas SMRT and ONT, these are real time observations, right. So, again if real time observation is important for your experiment, you want to consider SMRT and ONT. Okay. Throughput, again coming to all these platforms now, we can adjust this throughput because we have these different chips or different kind of sequencers, right, to cater to different throughputs, right. Now, there are few things that we need to first consider before we can answer that question, right, which NGS platform is most suitable for my experiment. Okay. So, the first 2 points, of course, the type of experiment that you are doing, the end goal of that experiment, right. Then of course, what is the read length that you need, the accuracy that you need, and the coverage that you require, and of course, the funds available, right, how much money you have. Okay. So, all these things you need to carefully think about before you can decide on the sequencing platform. Okay. Type of experiments, these are the type of experiments that you can run, right. So, you might be doing genome sequencing or transcriptome analysis, you can look at epigenetic modifications or metagenomic studies, or you can do something called amplicon sequencing, which is sequencing PCR products, right. You have only a small region of the genome that you are interested in, you are just amplifying that out by PCR and you are sequencing that region. So, this is called amplicon sequencing. Okay. Now, again for each of these types of experiments, you can have different end goals. Okay. So, for example, for genome sequencing, you have de novo assembly, you might be interested in sequencing a new genome, right, where you will do the assembly. You can have identification of SNPs and indels, right, you are studying a population and looking at genetic variation, for example, or you might be interested in structural variations, right, recombinations, rearrangement, gene fusions, etc. Again, for transcriptome sequencing, the most common goal is to measure expression levels of genes, right. So, this is the one common goal, but then you can have other goals also, right. For example, you want to study the alternative splicing events, or you might be interested in looking at difference in expression of different alleles of the same gene, right. So, in diploid organism, we have these 2 different alleles, let us say, of the same gene, and you want to see how differently they are expressed, right, whether they are expressed at the same level or there is some difference in expression of those alleles, right. So, these kind of studies you might be doing also. Again, epigenomics, you have methylation patterns, you can look at histone modifications or transcription factor binding, right, these are different aspects. And one of the key questions for all of these projects, right, is your experiment genome wide or targeted to specific regions of the genome, because this will again determine whether which platform you use, whether you use Illumina, you use long read sequencing, etc. right. So, these are the questions, these are the aspects that you need to think about carefully, right, and I will give you some examples, right, and that to help you out with this process, right. So, this is first example, how do you choose the best experimental design for your project, right. So, let us take this scenario where what is required is that we want to do a de novo assembly of a genome from a new organism, right, which has not been sequenced before, and we want all the information on single nucleotide polymorphisms, insertion, deletions, and structural variants, right, for comparison with a related species, right. So, we want to do the genome sequencing, and we also accurately want to determine these differences compared to a related species. Okay. So, here the assembly problem is there, but we also want to add this mutation detection problem, right. So, you need long read sequencing. So, what would be the solution here? So, the solution is you can probably use long read sequencing plus short read sequencing. You can use this combination of both, because long read which may not be like traditional long read methods, which may not be very accurate, right, but they can give you the structural variant information, right, and they can help in genome assembly process. Whereas short reads, they are much more accurate, as we have seen. So, they will be more useful for this single nucleotide polymorphism study or indel study, right. So, this is one solution. The other solution, if you have access to high accuracy long read sequencing, right. So, as we have talked about, right, for example, hi-fi reads in SMRT or in the consensus sequencing in nanopore technology. right. So, this is the solution. Let us take another example, right. So, choosing the most appropriate experiment design, right. So, here in this thought experiment, right, we want to do a deep sequencing of a 1 kb region, okay. And why do you want to do deep sequencing? You know what is deep sequencing, right, coverage of 1000 or maybe more of this region for identification of rare variants, right. For example, we are looking at a very important gene, right, in human, and these variations in this gene can lead to certain disease or affect certain metabolites, right, etc., that can lead to disease, right. So, this is something we want to study. Now, the answer here is not really straightforward, okay. So, one of the questions that you might have, that you want to ask still, is that where do you expect these variations to occur, okay. Are these variations, so I will draw this here. So, are these variations near the end of this 1 kb region, okay. Do you expect these rare variants to occur only between, let us say, last 100 base pair here and first 100 base pair here, right, of this region, or do you expect these variants to occur throughout the genome, throughout this region, 1 kb region, right. So, not just at the end, but throughout this 1 kb fragment, okay. So, these two cases will have two different solutions, okay. It is not just one solution. So, in the first case here, this one, right, since they are at the towards the end, right, end of this region, right, and the start of this region, right. So, you can actually reach these regions with short reach sequencing, okay, and perhaps then you can do something called paired end, right. We have discussed this paired end sequencing. You can do paired end sequencing perhaps in Illumina platform. Maybe this is a solution, okay. Right. So, this might be one solution, but then again you might have to be a bit careful about this fragment size. So, Illumina might be able to take only certain fragment sizes for this cluster generation or bridge amplification, okay. So, this is something you again need to inquire or consult with the literature and the Illumina sequencing platform literature. In the second case, here you cannot go ahead with this paired end sequencing, right, because these mutations are scattered throughout this 1 kb region, okay. So, what you have to do is either you have to go through this long read sequencing, right. So, either use nanopore platform or SMRT sequencing and use the consensus call, right, CCS reads in SMRT or consensus call in nanopore sequencing, right. So, as you can see, this requires a lot of thought, right. First you need to know your requirements, right, and then you can think about what kind of solution would be the most appropriate for you, okay, and what these technologies are capable of. Okay, so, the third and final example, right. So, here we want to do genome wide quantification of gene expression and we want to discover splice variants, okay. This is a very interesting example, right. So, we have, let us say, we are working with a species which has around 5000 genes, okay, and we want to measure expression level of all these genes, and also we want to look at splice variants, right. So, you understand what are splice variants, right. You have this exon intron structure, right, and they can combine in any combination, right. So, the splice variants you can have this E1, E2, E3, right. This is combine all exons. You can have E1, E2 only, or you can have E1, E3, right. So, whether you get this E1, E3 or not. So, how do you actually find out, right, whether this? So, here just maybe doing the single end sequencing may not be good enough, and probably you want to do paired end sequencing, right, because if pair end, if you see, for example, in this scenario, if you have E1, E3 combination and you get these reads, one of which falls on E1, the other one falls on E3, right, and they are from in the same DNA fragment, right, then it will tell you, okay, this is probably a splice variant, right. This is an E1, E3 splice variant. This is a different combination than the having all these exons in there. Okay, so that concludes our discussion. These are the references that we have used, and just to conclude, right. So, we discussed, right, sequencing coverage is an important metric for choosing the right NGS platform, right. You can play around with read length, the etc., the coverage, etc., and decide on the right platform. The quality score we have discussed, this gives us an expected error rate which could be used for downstream analysis, right. We will see, we will incorporate these quality scores later on in subsequent classes and see how that is important. And finally, we talked about NGS experiment design. So, designing an NGS experiment requires careful consideration of many features, right, and depending on your need and depending on the availability, you would find the optimal solution. Okay. Thank you.